First step of meiosis is interphase. And during interphase, the cell grows and copies. So
Copies of DNA.
we're going to add this.
The DNA is the yellow and we use the yellow and green pipe cleaners as the DNA, so the DNA is copied.
And then the centrosomes also enter the cell. That's it.
That's the end of interphase 1, or just
The
interphase.
second phase of meiosis is called prophase 1. So during prophase 1, what happens is the cell grows even more. So we're going to expand this out a little more. That's a little weird shape, but that's fine. And the nucleus breaks down. Before it breaks down, I was correct. The chromosomes condense into pairs like this. Sorry. And after they condense, they find two of the same size, so these two larger ones, and they pair up just like this. So, and that is, it doesn't actually look like that, but that's the end of prophase 1. I forgot to mention that the centrosomes line up at the top and bottom of the cell just like that. Then at the start of metaphase 1, the homologous pairs line up in the center on a thing called the metaphase plate. And on the metaphase plate, two spindle fibers connected from the centrosomes connect to the homologous pairs just like this. Alrighty. Oh, that's a little too far, but that's fine. Just like that. And that's the end of metaphase 1.
Next phase of meiosis is anaphase 1. In anaphase 1, the spindle fibers shorten, pulling each homologous pair down to the opposite poles. Sorry about that. So each one of these is a sister chromatid, and they pull down to the opposite poles. That's the end of anaphase 1. After that, the next stage of meiosis is telophase 1 and cytokinesis. During telophase 1 cytokinesis, the nuclear membrane reforms around the pairs of chromosomes, like so. And the DNA is unfolded. So the DNA will unfold. Shoot, we're going to cut that out. It's fine. After that, the contractile ring will come in and it will shrink away and pull these two together to create two different cells. Meiosis is prophase 2. In prophase 2, nucleus breaks down and the DNA condenses into chromosomes. The centrosomes then go to the left and right opposite poles and spindles form between them. That's the end of prophase 2. Alright, so the next phase of meiosis is metaphase 2. In metaphase 2, the chromosomes line up on the metaphase plate. After they are lined up, the spindle fibers will pull each sister chromatid to the opposite poles, like so. That is the end of metaphase 2. In meiosis, anaphase 2. Last phase in metaphase 2, I said that the spindle fibers pull the chromatids away, but that was actually incorrect. In the spindle fibers pull the sister chromatids to opposite poles in anaphase 2, not metaphase 2. And that is all that happens in anaphase 2. Last phase of meiosis, telophase 2. In telophase 2, the DNA or the chromosomes unwind and the nucleus reforms around them. And then after that, the contractile ring, which is made up of proteins, forms between the two cells, and it splits these off. I can't make that fit. I'll split these two. All 4 of them. In the nucleus into 4 sperm cells. 4 male sperm cells. So at the end of telophase 2, the 4 remaining, well, not cells, but they are actually spermatids, which then are formed into male sperm cells.